Gather round, guys. It's about that time. Time to tell you a story. Well, it all begins in the cytoplasm, where two molecules of ATP decide that they would take this first step towards breaking down glucose. You know what glucose is, that molecule that provides most of the energy in your bodies. So these brave molecules will attempt to break new ground, or what is referred to as the energy investment. Oh, yeah, okay. The first of these great pioneers kick glycolysis off by transforming an ordinary glucose molecule into fructose 1,6-biphosphate. However, this doesn't mean that our product has stopped being so changeable. What that means for us is that in a matter of seconds, fructose 1,6-biphosphate is cleaved to become what we're going to call G3P and DHAP. For the sake of uniformity, however, that DHAP is rearranged to form another G3P. So, inorganic phosphates are added onto our twin G3Ps, and two molecules of NAD plus are reduced. Remember those phosphates that just hitched a ride with our G3Ps? Well, they're delivered to two standby molecules of ADP, aka the less glamorous and adventurous settlers that followed our bold pioneers. Remember them? The two ATPs that kicked this whole thing off? So, these ADPs use these phosphate groups as a vehicle to become two molecules of ATP. This process is called substrate level phosphorylation because you add phosphate groups. The remaining phosphates the G3Ps still have are moved over to the middle carbons of the molecule, and water is formed as a waste product of that reaction. This is in the end, my friends, because these phosphates that are attached to our beloved carbons are once again employed in the grand transformation of two standby ADP molecules from drab to fab and full of energy. Yes, ladies and gentlemen, indeed at the end of the story of glycolysis, we are left with 4 ATPs. Yeah, 4! Where 2 had previously dared tread. In addition, our remaining carbons become pyruvic acid. Well, now that glycolysis is complete, you've only just actually started, so allow me to suggest 2 options. Pick your own ending story, if you will. On one hand, anaerobic cellular respiration, and on the other, aerobic cellular respiration. Anaerobic, you say? Interesting choice, actually. Okay, let me tell you what happens. In the absence of their dear friend oxygen, the pyruvates that resulted from glycolysis are unable to get into the oh-so-exclusive aerobic party, so they have to settle for the less glamorous, less efficient process of fermentation. Remember, guys, there ain't no party like an oxy party. But honestly, fermentation is like that one party where the DJ only plays that one song. You know, the party, we've all been to that party. Well, all fermentation knows how to play is glycolysis, so all you get is that same 2 ATP gain over and over again. So to keep glycolysis replaying in an endless loop, the cell needs more NAD+. So the only thing fermentation does is free up the NAD+ for the cell to use. In some organisms, like yeast, fermentation produces the ever-popular ethyl alcohol. No, okay. Bad news for the perpetually intoxicated, and good news for the rest of us that actually want clean livers. Fermentation in humans and most other mammals produces lactic acid instead of ethyl alcohol. So you made 2 molecules of ATP. Lame. How about if we had taken that other option, aerobic cellular respiration? Let me tell you what happens. In a more efficient world, our party-going pyruvates would have met up with their pal oxygen and headed out to the pre-party, more commonly known as synthesis of acetyl-CoA, but tomato. The pyruvates go in as themselves and come out as 2 molecules of acetic acid and 2 of NADH, an electron carrier. Remember that, it's important. Also, carbon dioxide is a totally wasted friend. Get it? Waste product, wasted friend. It gets kicked out of the cell. You exhale it out. Their next stop is the mitochondrial matrix if it's a eukaryotic cell, or just another part of the cytoplasm if it's a prokaryotic cell, where the energy from the pre-party, the acetyl-CoA bonds, is put to good use, creating 2 FADH, 6 NADH, and wonder of wonders, 2 molecules of ATP, ladies and gentlemen. And of course, you have the obligatory waste product, CO2. So finally, we have reached the last step of aerobic respiration, the electron transport chain. Yeah, what is the electron transport chain, you ask? Well, I can tell you. The electron transport chain is the collection of molecules embedded in the inner membrane of the mitochondria in eukaryotic cells. Electron carriers alternate between reduced and oxidized states as they accept and donate electrons. Electrons from the chain come from NADH and FADH too. I told you, don't forget that NADH, it's an electron carrier. while the final electron acceptor is a good old oxygen, oxygen, our pal. Each oxygen picks up a pair of hydrogen ions to form water. Water, therefore, is a byproduct of the electron transport chain. The question is, who's more powerful, NADH or FADH? Well, NADH provides 3 times more energy for ATP synthesis than its partner FADH. Who's important now? NADH. I told you. So why hasn't any ATP been produced yet? Because chemiosmosis hasn't taken place yet. Duh. ATP synthesis uses the energy of an existing ion gradient to power ATP synthesis. ATP synthase is actually like a rotary motor. It has 4 main parts, each made up of multiple polypeptides. Protons move one by one into binding sites on one of the parts, causing it to spin in a way that catalyzes ATP production from ADP in inorganic phosphate. So all of the hydrogen ions in the intermembrane space now go where? Through ATP synthesis. These hydrogen ions phosphorylate ADP, and the result is ATP, about 32 to 34 ATPs to be exact. And if you add the 2 ATPs that we produce in the Krebs cycle, you get like 36 to 38 ATPs. Can you believe that? Glycolysis, acetyl-CoA, synthesis of acetyl-CoA, Krebs cycle, electron transport chain, all parts of the aerobic cellular respiration, and we end up with 36 to 38 ATPs. In comparison, anaerobic cellular respiration, fermentation, remember that party? Totally lame. Only got you like 2. Which is the better choice? You know what it is. So how many ATPs does it take to make a snowman dance? About 38.